Hi, everyone. Thank you for coming to the HD Research Update session. I'm happy to see so many of you here after those other amazing sessions that we've had throughout the day. So my name is Rachel Thompson, for those who don't know, and I am the Manager of Development here at HSC. I'm very pleased to introduce the speakers for our next panel focusing on HD research. So today we have Dr. Rachel Harding, who is an HDSA Berman Topper Career Development Fellow working at the Structural Genomics University of Toronto. And Dr. Harding researches the 3D structure and shape of the Huntington protein to try and understand how this big complex molecule works and how the Huntington disease mutation might affect it. And then we also have Dr. Tam Maiuri. She is a research associate in Dr. Truant's group at McMaster University. So the Truant lab studies the normal biological functions of the Huntington protein in hopes of understanding the Huntington gene that causes HD may disrupt these functions. Dr. Maiuri is investigating the role of the Huntington protein in the DNA repair process in a project funded by the HDSA Berman Topper Fellowship. So I know everyone is quite excited for this session, and I'm not the only one looking forward to this presentation. It promises to be both fun and educational, so I will hand it over to Dr. Maiuri and Dr. Harding to get started.
Great, thanks. So Rachel's going to share her screen. All right, looks like we're ready to go. Thanks so much for the invitation to come speak with you about the HD research, an update. Rachel and I are always super happy to talk to people about what's going on in the HD research world. And we decided to have a little fun with this presentation. So you'll see it's called HD Research and the Quest for a Cure. And we've got Harry Potter on the front going after the golden snitch. So hopefully most of you are familiar with Harry Potter, but we thought maybe before we start, we should have a poll to make sure. So if you could, I don't know if you guys can see the poll just yet. We'll give you a chance to vote. So the question is, how familiar are you with Harry Potter? And the answers are, I learned more from Hogwarts than I did from high school, or I saw the movies or read the books a long time ago, or who is Harry Potter? Which so far we have one vote. All right, here they come. All right, well, we'll just wait for these to tick in. This will give us a little bit of an idea of how, how many people are going to get the more subtle references to Harry Potter, and how many people are just going to think that we're absolutely ridiculous for saying the things we're saying. So either way, I hope that everybody can have some fun with this and learns a little bit from this session. All right, we'll just give it another second to tick up. We've got 37% of people are experts. Yes. 37% are going to be at least familiar, and about a quarter of you have no idea, and feel free to laugh. All right, Rachel, do you want to do a quick introduction?
Sure. Yeah. So it's really great to be here with you guys today. For those of you who don't know me, my name's Rachel Harding, and I think I would probably be in Gryffindor. But what I actually do in real life is I work in a lab at the University of Toronto. Very close actually to where Tam works, and Tam and I actually collaborate on some different projects. So it's really fun to be working here together with her. Right, Tam, do you want to jump back to you now?
Sure, yes. Want to forward to the next slide. I have a wand here that I'm going to use to magically forward the slides because I don't have control of the screen. So that worked good. So far, so good. All right. So I always like to start these talks with a little family history because HD is a family disease, and I'm sure that this story will you'll be able to relate. A lot of you will be able to relate to such a story. So on the left-hand side of the screen, you can see a very old-timey picture. That's my great-grandparents with two of their 4 children. One of their children was my grandfather, who's pictured in the middle there. And unfortunately, my great-grandfather had the HD gene and passed it on to all 4 of his kids. So on the right-hand side, you can see me as a 2-year-old with my great-grandma Pearl, who lived to be 101. And she, of course, outlived her husband and all of her children, having lost them to HD. So also depicted here is my mom in the 80s. And we really didn't know whether or not she was affected. And Clicarius. We learned much later that she did in fact carry the gene. She was tested. She wasn't showing symptoms in her early 50s, so we really thought she was in the clear. But she got tested. She tested positive, and it was at the end of my graduate work that we found this out. So I was studying cancer. I was doing cancer research, and at that time, I basically switched fields and started working in HD research. So that's when I joined the Ray Truant Laboratory of Witchcraft and Wizardry. And I found there that, as Rachel Thompson explained, in the Truant Lab, we really try to look at the normal function of the Huntington protein, and so that we can try to understand what might be going wrong when the Huntington protein is expanded in HD. And I just want to check something really quick. Do we have Katie Newman on the call? I'm not sure if Katie's on the call, but I'm super happy to say that Katie Newman, who's many of you from YPAD will know, joined our lab now. And so Katie has many talents, and one of them is science. So we're super happy to have her on the team. Okay, so you may be wondering, what do we mean by the Huntington protein? So the next poll question is to gauge how familiar are you with the Huntington gene and the Huntington protein. So the possible answers are, I would ace a newt, which is also known as a nastily exhausting wizarding test on the topic. I've heard of the Huntington gene and protein, but I could use a refresher. Or when I hear the word protein, the only thing that comes to mind is the Canada Food Guide. So let's give it a second for the answers to file in. Okay, looks like most people have at least heard about the gene and the protein, with more of you requiring a little refresher. If there are people, if this is your first YPAD day and you've never heard a research update before, you may have never heard about a Huntington protein, and that's okay too. We will fill you in. Great. Okay, so next slideo. Let's start with the gene inheritance. So all of our genes are made up of DNA. And everyone in the world, everyone in the Muggle world and everyone in the magical world has a Huntington gene. There's a little stretch of DNA in the Huntington gene that's called the CAG repeat. And when that CAG repeat is expanded, that's when what causes Huntington's disease. So not only does everyone in the world have a Huntington gene, they actually have 2 copies, because we have 2 copies of every gene in our body, except in our sperm and eggs, where the copies get separated. So in this example, if the Potter family was an HD family and James Potter carried the CAG expansion in his Huntington gene, half of his sperm would carry the expanded version of the gene and half of his sperm would carry the normal version. And that gives Harry a 50-50 chance of inheriting the expanded version that causes HD, like the flip of a golden galleon. Progressarius. So now let's talk about how we get from the Huntington gene to the Huntington protein. So like I mentioned, genes are made up of DNA. And you can think of DNA as the recipe for all of the proteins that need to be made in the cell. Now, the proteins are actually the molecular machines that carry out all of the jobs in our cells and make our cells function properly. So the DNA is the recipe for the protein product, but there's an important step in between, which is that a copy of the gene is made, and that's the RNA copy. And there's a very good reason for this. Clickorama. There's a good reason for making a copy of the original recipe. So you can think of it as the original recipe is, very precious, and we don't want anything bad to happen to it. So we don't want it to leave the library or the nucleus of the cell. And so what the cells do is they make a disposable copy of the recipe that goes out into the cell, and that's what the protein product is translated from, it's made from. And so in this way, we can keep the original recipes safe. Okay. Thank you. In the case of the unexpanded Huntington gene, then you're going to make an unexpanded RNA copy, and that will form the normal Huntington protein. And the normal Huntington protein will be able to function properly and support healthy cells and neurons. In the case of the expanded Huntington gene, you're going to end up with an expanded Huntington protein. It just has like a little extra bit on it. But somehow this interferes with the function of the protein, and that's what causes Huntington's disease. So when I say the function of protein, what do we mean exactly? Subsequentius slidius. So if we're trying to think about what the Huntington protein does, this is what we think about a lot in the Truant Lab, and we're trying to figure out how it might be gone wrong in HD. And, Rachel's work actually looks at this too. Rachel's looking at the fine details of the shape of the protein, and that gives us clues also about what it does. So what it does is it acts as it's a very large protein, and it acts as a scaffold, as a sort of hub to bring other proteins into proximity of each other. So it's also very versatile. So it can interact with one group of proteins in one situation, and then it can, change its shape slightly, interact with a different set of proteins to do a different job. So it's really a very versatile tool, much like a wand, that casts the appropriate spell at the appropriate time. Forward us. So, figuring out how the normal protein and the expanded protein are different is really a tough question. Next slide, yes. So the question really is, how can we fix it? So what we do know is that there's something about this expanded Huntington protein that over time, over a lifetime, causes brain cells to die. Now, thanks to some major technological advances in the last, decade or even longer, there are now ways to stop production of the toxic protein. And even more recently, there's ways to stop production of a super toxic version of the protein, which results from something called somatic expansion, which we're going to explain in a few slides. But we can also aim to treat the different types of damage that are occurring, such as inflammation. So there's a few different strategies of what we can do with the knowledge we have. And I'm going to hand it over to Rachel to explain how we can use an immobilis spell to stop production of the protein.
Thanks very much, Tam. We haven't actually rehearsed all those slide transitions, so I really appreciated all your spell knowledge in PowerPoint. It's great. So as Tam mentioned earlier, the kind of fundamental understanding that we have of biochemistry is that we have DNA that encodes the recipes to make different proteins. And in order for these recipes to be made, we have to make a copy of the recipe in this special molecule called RNA. And then this RNA tells other machinery in our cells how to make very specific protein molecules. So one of the ways that people have been thinking about for a long time is that it's quite difficult to get rid of a protein once it's already been made. But what we can do is interfere with this step. So we stop the RNA being able to be made into protein. And if we get rid of the RNA to be like a functional form of the RNA, then we'll end up getting rid of the Huntington protein. And so this overall strategy is often called Huntington lowering therapies. And that's what you'll read about a lot on different like research websites about Huntington's disease. And it's what you'd hear in the news a lot when people are talking about clinical trials. They'll often call them Huntington lowering clinical trials. And one of the main approaches that people have been taking for a little while now is to use these special type of drugs, which are called antisense oligonucleotides, which is a bit of a mouthful. So often people will just call them ASOs. And so what these do is they are special small pieces of nucleic acid, and they will come along and they will bind onto the message or the copy of the recipe to make the Huntington protein. And when they do this, they block the production of the protein and the levels of the protein will go down. And so you've probably heard about this in the context of quite a few different companies who have been making ASOs to lower the level of the Huntington protein in HD patients. Unfortunately, for some of these companies, this has been the result of some really sad and disappointing news. So one of the largest clinical trials that happened that was going until the beginning of this year was run by the drug company Roche. And they were looking at their ASO, which targets the Huntington protein, which is called tominersen. And very sadly, kind of towards the end of March this year, they had to halt dosing of the drugs. That means that the clinical trial itself hasn't stopped, but all patients in the trial stopped receiving the drug. And why was this? Well, one thing we know is this tominersen, this antisense oligonucleotide or ASO, the more of it you give to someone, the more it will lower the levels of the Huntington protein. So that seemed like it was doing what we wanted, and that was really good news. But then we subsequently found out from this clinical trial that actually the more drug you gave to people, so people who were receiving the drug every 2 months compared to those who got a placebo, which is like a fake version of the drug, or they were getting the drug only every 4 months, they were actually getting worse over time. So their symptoms, their HD symptoms were actually getting worse. And when bad things happen, often they all happen at the same time. So we also got this really sad news from another company called Wave. They also have an antisense oligonucleotide drug that was in a trial called Precision HD. And although their drug didn't make anyone get any more sick, it also didn't do anything. So here you're giving someone more and more drug as you move along. They either got 2, 4, 8, 16, or 32 milligrams of the drug, and it didn't actually do anything. So this was also really, really disappointing. And I think everyone was feeling very sad at the beginning of this year. And, you know, that sadness was felt not only, you know, like in the research community, but also by the people working at these companies and far beyond that. But what I really like about this quote is that even when things are really not great, there is always hope, and there's always something that we can get, some good that can come out of a situation like this. So this quote from Albus Dumbledore is that happiness can be found even in the darkest of times, if only one remembers to turn on the lights. And so that is what is happening right now. So when the trial was halted by Roche, this was a very sudden decision, because as soon as they realized what was happening, they have to stop everything really quickly. And unfortunately, because of the way the stock market works and other bits and pieces, no one had any pre-warning. So it just happened very dramatically. But that meant that there was then this huge logistical operation that needed to happen. So all these centers all over the world where people were participating in this clinical trial, we had to suddenly stop everything, collect all of the data, collect all of their samples, because they're collecting blood and also spinal fluid, and also all the information about those patients and their assessments from their doctors. And that all had to be done very quickly. And then everything needed to be shipped back to be analyzed, and the data needed to be crunched. And you might say, well, if the drug wasn't working very well and it was making people more sick, then why is this important? And well, that's because when we think back to seeing people not being as well, not doing as good when they receive the drug, that's like the average of everyone who is in the clinical trial. But there's still some important things that we need to work out. One of them is maybe some patients in the clinical trial were actually getting better, but depending on what stage of Huntington's disease they are at, this will change how the drug works. So maybe people who are younger, tominersen actually was doing them good. And we know, like, anecdotally that some people said that they were in the trial and they felt a lot better. So now what we need to do is look at all that data and work out, is that really the case? Another thing was that when they went from the very successful clinical trial, which showed that the levels of Huntington were dropping, to the most recent one, which had to be halted, they upped the level of drug given to patients quite dramatically. So maybe this was just too much, because something we know now from other clinical trials where they use these special molecules, these ASOs, is that even no matter what protein or what they're trying to target, this could be in something completely unrelated to Huntington's disease, we actually see some of these same negative side effects. So it could be something fundamental to the chemistry of that type of drug, not what it's actually doing, that is making people more sick. And the other thing that they'll want to do is work out, okay, if we look at a patient, how old they are, what is their CAG length, what other things are going on with their medical history, can we predict their response to the drug? So all of these things are really important, because there are a lot of other companies working to also lower Huntington with different types of drugs. And this will help inform them how to design better experiments, how to make their clinical trials more successful. And Roche is working really hard with so many people in the community to try and get us answers to all of these questions in early 2022. So we're going to be keeping our ears to the ground for that, and we'll be reporting on that in places like HDBuzz and through HSC as well. So Wave was a slightly different situation. So this is their drug just didn't work. And what they've done is they went back to the drawing board. Fundamentally, their idea is really good. An antisense oligonucleotide should work nicely to lower the levels of Huntington, but they've redesigned it. So they've now made a version of their drug which has much, much better chemistry. The other thing to note about Wave's drug is it's quite different to the one of Roche. So in the Roche trial, you're lowering both versions of the Huntington protein. So we all, as Tam mentioned earlier, we either have an unexpanded and an expanded version if you're an HD patient or someone with HD. So you'll be making both the expanded and the unexpanded form of Huntington. Whereas, so for Roche, you would be lowering both of the levels of those proteins. However, for Wave, they have this really clever technology, which means that you're only going to lower the toxic form of the Huntington protein, so this expanded form of the protein. And so the way that they do that is that they will look for very specific signatures in your DNA code that are only found in the toxic Huntington gene. So this means that patients have to be screened to make sure they have those signatures before they can take any of Wave's drugs. But it means that you should have something very specific. So you're only going to be getting rid of the toxic protein, which should, maybe that will be what will make the difference. And so this new drug that Wave have is called Wave 3. And it's going to be tested in a clinical trial called Select HD, which should be starting next year, they believe. But in the meantime, what they've been doing is a ton of experiments using this new version of the drug in cells in a dish or in mice, and they also have done some work in monkeys. And they presented a lot of this work very recently at a scientific meeting, and all the results look very encouraging. So we're very hopeful that this new clinical trial will give us more promising results. So there are other ways that we can lower the levels of the pesky Huntington protein, and one of those is using gene therapy. So gene therapy is an irreversible treatment that basically means that once you've been treated with it once, it's a one-shot therapy that will maybe cure you, but the idea is that it's one shot and then that's your treatment for life. And we hope that that will make people better. And so the idea behind this gene therapy is that you have a special form of a harmless virus that contains instructions within it. And what happens is those instructions get stuck in your own genetic code, and they allow your body to make essentially something very similar to an ASO that will block and bind the RNA. And when that happens, you will also lower the levels of the Huntington protein. So you get rid of the RNA, the recipe for the Huntington protein, and then you get rid of the protein itself. And because this is a one-shot experiment, procedure, it's being done by this company called uniQure. And the way that they're doing it is rather amazing, but a little bit scary, where you'll have an injection. Patients receiving this will get an injection in the brain to deliver this harmless form of virus into the brain. But then that's it. So it's quite scary to think about this as a procedure, but if it works, then that's that. And it will be really, really interesting to see how that goes. So this is a bit like when Ron is talking to Hermione, and you're a little scary sometimes, you know that, but brilliant. So this is amazing science and incredible technology. And we're incredibly grateful to the very first patients. There was 2 patients who received this drug, and they volunteered themselves in the very first safety study, and they're incredibly brave, wonderful people. And because of their bravery, we've gone on to learn lots and lots of different things that's happening. uniQure has been testing this drug more and more in lots of different types of animals, as well as monitoring the people who originally received it. And again, it looks very promising. And that's amazing that this technology has allowed them to develop a drug like this. And what's great now is that this is now going into clinical trial. So this rather scary but amazing technology is going to be used in 2 trials, one of which will be in the US and one of which will be in Europe. And this is going to see further test the safety of this new drug in people, but then also see if people get better when they've received the drug against a few different criteria. All right, so another way that people are trying to lower the Huntington levels is by targeting this process that happens in cells in our body called splicing. And so, as you're probably all experts on now, we have the DNA, which allows us to make a copy of specific genes, which makes a molecule called RNA. But before that RNA can be turned into a protein, there's actually a lot of things that need to happen to that RNA molecule. And that process is called splicing. And so what that means is the RNA is processed through this procedure called splicing, which is basically like chopping up the RNA with a pair of scissors. And it will remove certain sections of the RNA so that the protein can be made from this new form of the RNA molecule. But what people have found is that there's this really cool new type of drug which will target this procedure, splicing. And it means that the RNA can't be chopped up. And when that happens, you actually can't make the protein. And so these drugs are called splicing modulators. And this is a really interesting new area of technology, especially in the field of Huntington's disease. And yeah, so as Minerva McGonagall would say, I've always wanted to use that spell. And now we've got the technology to exploit this part of the process, which means that we can hopefully lower Huntington. And just as soon as anyone has one good idea, someone else comes along with the same idea. And that's exactly what happened in this instance. So there are two companies. Novartis is one of the biggest drug companies in the world. And then there's this smaller company called PTC. But they've basically come up with splicing modulators, these special types of drug, which have been shown in different systems in the lab to lower the levels of Huntington. But there are two different drugs. One of them is called branaplam. The other one is called PTC 518. And these cool splicing modulators that lower Huntington will hopefully be tested in clinical trials very soon. So this is really exciting to see this cool technology be used in this way. All right. And with that, I'm going to hand it back to Tam.
Thanks, Rachel. So yeah, so this Huntington lowering business is super exciting, and there's many approaches, as Rachel explained, but it's not the only form of magic being used to address potential therapies for HD. So we're just going to go through one more. And for this, have to explain this important finding from a few years ago, which really came about because the researchers were asking the question of when symptoms start, or more specifically, they're asking the question, why do symptoms start earlier in some people than others? And, you know, we knew that the length of the CAG repeat matters. So in general, the longer the CAG repeats, the earlier people get symptoms, but there's just a huge amount of variability from person to person. And so what this study was addressing was, out of all the other genes in the body, what genes might influence whether people get symptoms earlier in life or later in life. And so what they found, these would be called genetic modifiers because they're basically genes that modify the disease. And what they found was that DNA repair genes made up a large proportion of the genetic modifiers that they found. And so this was super exciting because it basically gave researchers another strategy to develop drugs. You know, if DNA repair genes are influencing symptom onset by decades, then if we can make drugs that target those DNA repair proteins, then we could possibly influence symptom onset by decades, which would be so amazing. And so DNA repair, you know, it's not just a thing in HD. It's actually common to, problems with DNA repair are common to many neurodegenerative diseases. But there is something special about HD and DNA repair. Okay, now we can go to the next slide. And this is what I mentioned earlier. It's called somatic expansion. And how this works is basically that when we say somebody has 40 CAG repeats, what we're referring to is the number that they were born with. And so every cell in their body will have, their Huntington gene will have 40 CAG repeats in it. But what happens over time as people get older is that in some parts of the brain, some cells, the Huntington gene, the CAG repeat starts to expand further. And so you might have some cells in the brain that have 80 or 100 CAG repeats instead of the 40 that the rest of the body has. And of course, this is going to, in those cells, lead to the production of Huntington protein that's further expanded, and then later in life, very highly expanded. And presumably, this is more toxic than just the 40 repeat expansion. And so it's almost as if the CAG repeats were bewitched with Bellatrix Lestrange's Geminio curse, which basically causes objects to multiply repeatedly if they're touched. And who would cast such a spell? Well, it turns out that it's because of rogue DNA repair proteins. So these rogue DNA repair proteins, you know, they didn't mean to go rogue. they're really just trying to do their job and fix damaged DNA, which happens in the brain all the time. But there's something about, a 40 or a 42 or an expanded CAG repeat. There's something about the shape of that the DNA repair proteins just get mixed up, and they end up adding more CAG repeats instead of fixing the damage. Okay. Advanciarmus. So I hope that the people who have never heard of Harry Potter are having a good laugh at us. So there is a company called Triplet Therapeutics, and they are addressing this idea of somatic expansion. So they're basically they've designed a drug against one of these rogue DNA repair proteins. And the hope is that the drug will reverse somatic expansion in HD patients. And so far, they've shown that the drug is safe in mouse and monkey models of HD. And so hopefully it will also be safe in humans. And like the Roche and the Wave drugs, this one is also a difficult one to deliver. But instead of relying on lumbar puncture, which is what Wave and Roche had to do, Triplet actually has come up with a new delivery system. So it's an implanted catheter that will deliver the drug into the deep brain structures where HD is thought to occur, basically. So the clinical trial for this one is set to start next year. So again, keep your eyes peeled for updates through the HSC and on websites like HDBuzz. In the meantime, Triplet is conducting a trial called SHIELD HD. And this is just an observational trial. There's no drug involved. What they're doing is they're assessing how this somatic expansion happens over time in HD patients, just getting the information. And the point of this is that it'll actually help inform how to set up their trial, what patients might be most likely to respond to the drug, maybe when the drug should be administered, at what point in the course of disease. These kinds of questions can be answered by observational studies, because the observational studies kind of give us a chance to predict what might happen, much like looking into a crystal ball. And so that's what Triplet is up to right now. And SHIELD HD is just one of several observational studies. So there are some bigger trials happening. Enroll HD, you may have heard of. This is a very, very large trial, strictly observational. It's just collecting immense amounts of data that is super helpful for, companies when they're designing their trials. It's helpful even for basic researchers like us. If we have an idea, if we come across something in our experiments using cells in a dish, we can go to that data and ask, are people that are on a certain, there was one with metformin, that's a good example. There was some evidence that metformin might be helpful in HD animal models and in cells. And so those researchers were able to go and mine the data from Enroll HD and ask, out of all the people from Enroll HD, how many are on metformin, and how does their progression compare to people who aren't on metformin? And this amount of data is just hugely valuable. So what we need to know about Enroll, they're still recruiting all the time. You do not need to know your gene status to be part of the trial. There are sites all across Canada. So there's Enroll HD. There's also HD Clarity. HD Clarity is I didn't mention what they do for Enroll. That's what I wanted to say. I knew there was something else. They basically they do a few like cognitive tests, movement tests, and they take a vial of blood. It's basically like a 1 to 2 hour time of your day once a year. So for HD Clarity, a little bit different. This one is specifically taking spinal fluid samples. So that involves a lumbar puncture. For this trial, you do need to know your gene status. And the Toronto site is fully recruited, but the Vancouver site is still recruiting. So just to kind of sum up, we have many, many approaches happening. And I think what Rachel and I really wanted to, the message we really wanted to share with you today is that the problem of finding a treatment for HD, whether it's, getting right to the root of the problem, the Huntington protein, expanded Huntington protein, or whether it's treating symptoms and just improving quality of life. There are a lot of companies working on this, and there's a lot of different approaches. So here's, just a list of some other companies, 2 of which have trials currently in Canada. So Prolenia is running a trial called Proof HD, and Neurocrine Biosciences is running a trial called Connect HD. And Rachel's going to talk a little bit more about where to find information about trials in a bit. What's our next slide, Rachel?
Yeah, so our next slide is this last poll, which is how would you rate your knowledge of HD clinical trials? So we've been through quite a lot of information in a short amount of time. And you can find that poll should be available for you to answer, hopefully. So the answers are that you have a Hermione Granger level of knowledge and you're a living, breathing encyclopedia on HD clinical trials, or you're at the Ron Weasley level where you've got some basics, but maybe need to do a bit more reading, or Neville Longbottom where you're lost and there's just too much going on and where can you find out more and read more about this. So most people are Ron Weasley, which is excellent. That's absolutely great. It's good that people have got a little bit of understanding about how all this works. We've got a Hermione Granger in our midst as well. Excellent. Happy to see that. Awesome. So yeah, I mean, you're not expected to, we're not going to be testing you on anything that we've been presenting today, but what we do want to do is leave you with where you can read more about everything that we've been talking about today. So Tam and I, as I mentioned at the beginning, is that we both write for HDBuzz. And for those of you who are not familiar, this is a sort of plain language HD science and research news website. And on here, every time that there's a new paper published about a clinical trial or really interesting piece of science about Huntington's, we will do our best to write a plain language article that will cover all the latest and greatest news in Huntington's disease. And so most recently, a lot of the companies that we spoke about earlier, they actually all gave presentations at a conference called the European Huntington's Disease Network. And so there you can actually go through and there's like little summaries of what every single company is doing, what clinical trials they have, and you can read all of that information and the science behind like how all their drugs work as well. So I recommend reading that. And if you don't understand anything, you can always contact the editors and tell them that we need to do a better job at writing. The other thing is I would highly recommend going to both the HDSA, but also obviously the Huntington Society of Canada. Both have a lot of information on the different clinical trials that are ongoing for Huntington's disease. So HDSA have this very specific tool called HD Trial Finder, which can be very helpful. But this information is also available on the Huntington Society of Canada website. So you can see which studies are currently ongoing, which ones are recruiting, which ones are clinical, which ones are observational. And so if you want to get involved in a trial, this is a really great starting point. Okay, and finally, something that we just wanted to chat with everyone here is that, you know, we're very lucky in Canada to have such amazing access to COVID vaccines. And one question we get a lot at HDBuzz is asking about like whether people with HD or with HD in their families should get the COVID vaccine. And those of you who are unfamiliar, this is Professor Ed Wild. He is like one of the leading HD researchers in London in the UK. But he also spent most of his free time in the pandemic giving everyone their COVID shots. So he set up and ran multiple vaccination clinics across the city of London. And he would tell you, yes, get your shot, because people with HD are actually at higher risk from COVID. The vaccine is safe and effective, and we now have really great data on that because so many people have been vaccinated. But the important thing is that there's the vaccine won't stop you from being in any future clinical trials. If anything, it might become a requirement for some clinical trials. So we would just want to strongly encourage everyone to get their COVID vaccine as soon as they can. All right.
Great. So we just wanted to conclude by saying, I hope that we've convinced you that there's lots of magic happening in labs all over the world, coming up with ideas that big companies, big and small, are picking up on, and really starting to push through to real possible treatments. And so it's like a game of Quidditch. These companies are all on their broomsticks racing after a treatment for HD, just like they'd be racing after the golden snitch in Quidditch. And if you're not familiar with the game of Quidditch, the goal of the game is to catch the golden snitch. And the game does not end until the snitch is caught. So with that, thank you very much for having us, and we'd be happy to answer any questions.
Thank you, Tam and Rachel, for that wonderful presentation. We're now going to open it up to questions. So if you do have a specific question, please feel free to add it into the chat here and we will touch on it. It looks like we have one already. So the question is, does lifestyle and health habits contribute? I'm assuming it could be a question around clinical trials or COVID rates, but Jody, if you
I think it's around somatic expansion. So I think that's when I saw that pop into the chat. So yeah, that's a really great question. And so we know that somatic expansion is happening in HD patients, but what we don't know yet is exactly like precise details about who it's happening to faster, like when it happens, why it happens. And although we have some clues and some details about this process, there's still like a huge amount more that we need to learn. And so that's why the SHIELD HD natural history study is going to be so important, because they're going to try and answer this exact question. So by looking at this group of Huntington's, people with Huntington's disease, they're going to be looking at their rates of somatic expansion and seeing how that's going for them. And then we might be able to start answering some of these questions. But at the moment, we don't know if there are specific things that might be changing it beyond what we know about some of these genetic modifiers that Tam mentioned earlier.
Jody also added, yes, about the somatic expansion, but also whether lifestyle and health habits contribute to the onset and intensity of symptoms. And the answer to that is most likely. So I think that might have been when we were talking about how, some people get symptoms early and late in life, and that study looked for genes that contribute. There's a certain sort of percentage of the difference that can be accounted for by genes, but not 100%. So there are other factors, obviously, that are, playing into this. They're most likely to be environmental, there's going to be environmental factors. So I think that in general, the answer to this question is no matter what disease you have, in general, eating well and exercising is just going to help with whatever it is that ails you. And the same would be true for HD. Okay, does alcoholism progress the symptoms due to killing off brain cells and unhealthy cells? I don't know of a specific study that has proven this. Do you know, Rachel, of a specific study that's looked at this?
No, I don't. But I think this kind of answer goes back to the answer that you just gave, that although we don't have, like, for some very specific items or like drinks or food that you need to be eating or drinking, like how they might change things specifically. I think just generally living a healthy lifestyle and exercising has definitely been shown to be beneficial. Yeah. But I don't think there's been a specific study about alcohol intake and HD symptoms, to my knowledge.
Okay, we have another question here. Has the halting of dosing of Tam Maiuri generation HD1 impacted other clinical?
Sure, so I can take this one. So yeah, so the Roche clinical trial was halted for dosing, but the trial is still ongoing. And so what that means is that patients are no longer receiving the drug. They had the option to leave or stay in the trial, but what's really good is that 85% of people enrolled in the trial decided to stay in, and they're being monitored and data is being collected from them to see how they're doing and to check in with them and making sure that they're getting the care that they need. And so that's good. And so with that data, people are expecting that when we start crunching what's going on and how people were impacted by the trial, it will probably inform some of these other Huntington lowering trials that are happening. So for example, one of the things I mentioned earlier is that there seems to be something about these ASOs, these antisense oligonucleotides, that we've now seen in clinical trials for a number of diseases, not just Huntington's, but it's leading to these same negative symptoms, these poor symptoms, regardless of what the disease is and regardless of what this ASO is trying to target in your cells. So it could be something very like to do with the chemistry of an ASO that just causes this inflammation and like sort of poor symptoms in the brain. And it could have nothing to do with the Huntington lowering. So if finding out an answer to that question will be either good news or bad news for people who are also working on Huntington lowering, but using different types of drugs. So for example, like Unicure is doing Huntington lowering, but they're using this virus as the way to get the drug in to people. And so working out if it's something to do with the ASO chemistry or whether it's something that's bad about Huntington lowering will obviously have an impact in subsequent clinical trials. And so that's why we're all kind of on tenterhooks trying to wait for them to release that data. And hopefully they have sufficient data and evidence to like inform those kind of questions. So this is also why it's taken them so long to go through the data. So by the time they released this data to try and answer some of these questions, it would have been a year since the trial was halted. And I know that's a really difficult and long period of time for everyone who was involved in that trial. But the reason it takes so long is, number one, this huge logistical operation, but number two, because they have to be absolutely sure that they're clear on what the data is telling them because it will impact all these other clinical trials as well. So yeah, watch this space. And I know that they are planning to deliver all that information. There's a conference run by CHDI, who are a big funder of HD research, and they are planning to make their announcement at that conference that they run at the end of February. So we will have answers then, and HDBuzz will be live reporting all of that on Twitter, so you'll be able to see what's happening in real time. So we'll do our best to get information to people as quickly as we can.
Oh, you're on mute, Rachel.
We can't hear you, Rachel.
Sorry, I was going to say, we have a question. Is CRISPR a possibility to cure HD? We've heard that name before, but if you want to expand.
Sure, I can start this one. I'm sure Rachel will have something to add, probably. I'll miss something, but so just to explain what CRISPR is a gene editing tool. And so very, very amazing technology, recently Nobel Prize for the discoverers of this. CRISPR is used by labs all over the world to indirectly, find cures for HD. Whether or not CRISPR can directly, gene edit out the expanded CAG repeat, that is not possible at this time. So there's a few reasons for that, but probably the biggest is that, so there's kind of a few ways to think about this. So there's the idea of editing, before a person's even born. And that is really, I'd say, like, still in the realm of science fiction at this point. We're just not there. We're not there understanding all of the things that happen with CRISPR to be at the point where you could edit an embryo before it's born. CRISPR will be able to be used in people's tissues. For example, if you had a blood disease, they can take your blood out and use CRISPR to fix your genetic blood disease and maybe put your blood back in. That's a possibility for those types of diseases. In HD, it's harder because the disease is happening in the brain. So you can't really do that. But as technologies move forward, some of these things are going to come together. So the short answer is it's not a possibility right now, but in the future, it's likely to be. Rachel, what would you add to that?
Yeah, no, I think that's a really excellent summation, Tam. And I think, the key point is that there are diseases that that we know in principle can be cured with CRISPR nowadays, but those aren't neurological diseases. But the thing is that, like, CRISPR was only discovered I think 8 years ago now, and we've already come so far. So this technology that no one thought could ever exist was discovered within the last decade, and now it's already being used to treat a disease that no one thought could be treated in this way. And so there's what's really exciting about that is that all these new technologies, they're constantly being developed by hundreds of labs all over the world. And so these are really smart people who will come up with great ideas, and maybe, 5-10 years time, the possibility of using CRISPR to treat all sorts of different diseases might be very different, but we just need to watch that space and see what happens, and just be hopeful as these very smart people develop the technology to do more applications.
Okay, we only have a couple minutes left for questions, so we're going to rapid fire the last two. Is there any research on CBD or THC oil with HD happening?
I'm not sure. I'm not aware of any. Rachel, do you know?
I'm not aware of any specific clinical trials happening in patients. There are, certainly to the best of my knowledge, there aren't any in the registry that I've seen. But there might well be experiments that are being done on a smaller scale in the lab. But yeah, no, I don't have any information on that, I'm afraid.
Sorry.
Okay, and last question before we wrap. Are there any trials in particular ongoing for other diseases that might help with HD?
Yes, actually, the drug that Rachel talked about being developed by Novartis, one of the splice modulators, that was not originally intended for HD. That was intended for a different disease altogether called SMA, spinal muscular atrophy, which affects children. And it's effective in those children. And the good thing about this, so this is the drug called branaplam. The good thing about this is that we already know that it's safe in children. So it does need to be tested in adults too, but this is very promising. Can you think of any other examples, Rachel?
Name one. I mean, I would say some of the drugs that are being developed by companies for like more general movement disorders. So other diseases that cause very similar symptoms to Huntington's. Those are drugs that are being aimed at multiple different illnesses. And so that's really promising. So valbenazine is like one of those drugs. And so that's where we're learning a lot from other communities as well.
Great. Okay, one last question Jenna snuck in. Do we know where Triplet is considering doing their trial?
I asked this to Tiago today.
I can't remember right now. Do you know, Tam?
No, I don't. And I asked Tiago Mestre this very question today. He's a clinician who does run trials. And he said he had not been in Canada. And he said he had not been approached yet by Triplet. And that's all he knew. So I'm really hoping that there will be Canadian sites for that, but just keep your ears open for announcements. We'll make sure we get that information out ASAP.
All right. Well, thank you so much for this fascinating presentation and the overview of clinical trials and the pipeline to get to them. We really appreciate giving your time and expertise today. Make sure you stay connected with both Rachel and Tam through HDBuzz on their website or on their Twitter. They're always posting new updates that are very helpful for the community. And so now we're going to head over to the wrap-up session on InEvent here from our new board chair, Brandon. So thank you so much again, Dr. Maiuri and Dr. Harding, and enjoy the final session.
Thank you so much for having us.
That was great.